Welcome to a video tutorial of TISMO, a database of syngeneic mouse tumor models and cell lines. The website is divided into 7 components accessible from the top menu bar. The interactive components can also be accessed from these 4 buttons in the middle of the home page. I will be walking through each component in this video. In the Data Browser module, we have 2 main tabs, Data Summaries and Metadata. The Data Summary tab contains overview tables summarizing immune checkpoint blockade-treated tumor models, cytokine-treated in vitro cancer cell lines, and treatment-naive baseline samples. The ICB treatment table is grouped by tissue and cancer type. Tumor model and treatment details are provided as well as the number of studies. Samples are summarized according to response status. In the cytokine treatment tab is a similar table with cytokine-treated syngeneic cancer cell lines. Lastly, the baseline tab summarizes in vivo tumor and in vitro cell line samples that were specified as controls. In the Metadata tab, you can browse information on the syngeneic tumor models and cell lines in the database. Basic cell line information is provided in the Cell Line Metadata tab. Cell lines are grouped by cancer type. Details include any parent cell lines, the background strain of the mouse from which the tumor originated, and how the tumor originated. If available, we also include the year the cell line was created and the sex of the original mouse. From the In Vivo Sample Metadata tab, users can locate relevant tumor models from our collection, grouped by study. Fields include study ID, most of which will be the GSC ID, a study identification number for the GEO database, cancer type, and cell and mouse-specific aspects of the study design, such as any gene perturbations or treatments. We also annotated whether the sample was from a primary or metastatic tumor, the type of implantation, whether it was orthotopic, that is, a tumor transplanted to the normal organ site, for example, the mammary fat pad for a mammary cancer, or non-orthotopic, such as a subcutaneous implantation, and the site of implantation. If the sample was from a study with immune checkpoint blockade treatment, this will be noted in the ICB study field as either a baseline, control, sample, or by response status, R for responder, NR for non-responder, and unknown if this information was not provided. The final column lists the number of sample replicates for each design. Similar metadata information is available for in vitro cancer cell line samples. For each tab, a search bar is available to narrow down samples according to specific search criteria. Now I will go over the gene module. In the gene module, we can compare the expression of a specific gene across immune checkpoint blockade treatments and tumor models, before and after ICB treatment, and between responders and non-responders. Let's look at the beta-2 microglobulin gene, B2M, which encodes a component of the MHC class I molecule, important in antigen presentation to cytotoxic T cells. You can either select from the drop-down or type your gene of interest into the gene search bar. Select your ICB treatment from the treatment drop-down. Here I will just select all treatments. You can also select a specific tumor model of interest. Let's look at all models. Then we click Submit for results. Along the y-axis, we have a list of the different cohorts that meet the search criteria. The first component of the cohort name is the tumor model. The second part is the study ID, and the remaining parts of the name describe the ICB treatment and any other treatment or condition, such as time point. The number in the parentheses represent the number of samples in the cohort. On the x-axis, we have the log TPM value of the gene selected. Baseline control samples are colored in gray, non-responders are orange, and responders are green. The differences between groups are statistically evaluated by the Wold test using DE-seq2, and the color of the asterisk represents a significant difference between specific comparisons at a particular FDR cutoff. For example, EMT6 is a mammary cancer model. The study ID is GSE 107801. There are 20 samples in the cohort comprising baseline controls in grey and anti-PD-L1 treated samples. In this cohort, all anti-PD-L1 treated tumours responded to treatment. The green asterisk indicates that there is a significant difference in B2M between responders to anti-PD-L1 and the baseline control samples. An underscore to the asterisk indicates that the first condition of the comparison on the left, here the responders, have higher B2M expression. This figure can be downloaded as a JPEG, PDF, and the data to generate this figure can be downloaded in table format. Similarly, gene expression across cell lines can be compared across cytokine-treated in vitro samples. We will now go through the pathway module. The pathway module enables users to compare sample pathway enrichment between conditions. Pathways were curated from KEGG, the gene ontology including biological process, molecular function and cellular components, as well as Wikipathway Reactome and MSigDB's C7 immunologic signature. Select a pathway from one of the 7 sources and compare its score, which was characterized by single-sample GSEA, across ICB treatment and tumor models in the Pathway in Vivo tab, and across cytokine-treated cell lines in the Pathway in Vitro tab. Let's look at the MHC protein complex assembly pathway from the gene ontology's biological process after cytokine treatment in a few syngeneic cancer cell lines: a lung carcinoma cell line, a colorectal cell line, and a head and neck squamous cell carcinoma cell line. We then click the Submit button for results. Descriptions of the cohorts are on the y-axis, with the cell line as the first component of the name followed by the study ID. The number of samples in the cohort are in parentheses. The score of the selected pathway is on the x-axis. The color encodings for the box plots have baseline control samples in gray, interferon beta in blue, interferon gamma in green, and TNF alpha in orange. The differences between groups for cytokine-treated in vitro samples are statistically evaluated by the student's t-test. Asterisks on the right indicate a significant difference between groups. They are also color-coded according to the comparison. Similar to the other modules, if the asterisks are underscored, it means that the first condition of the comparison on the left has the larger value. As expected, we consistently observe a significant increase in MHC protein complex assembly score after interferon gamma and interferon beta stimulation across cohorts, but not from TNF alpha, where we only see a significant increase in 1 out of the 3 cohorts. If you have your own custom gene list, you can upload it in the appropriate tabs. Please download the provided sample file to use as a reference. This is the example pathway file. Your uploaded list will have up to 2 columns, the first column containing gene names, and the second optional column for gene weights. Please check the header and weighted pathway checkbox as required. Leaving the weighted pathway unchecked will just read in the first column of your list. Please also select the separator used in your file and whether your gene names are mouse or human gene names. Then you can select the ICB treatments and tumor models of interest and submit your query. Next, we will go over the infiltrates modules. The infiltrates module uses 6 algorithms to infer immune cell infiltration levels in tumor samples. These include Timer, XL, CyberSort, EPIC, QuantiSeq, and Murine MCP counter. The differences between groups are statistically evaluated by the Wilcoxon rank sum test. You can explore immune infiltration in immune checkpoint blockade treated or baseline tumor samples. Let's use TISMO to look at CD8 positive T cell infiltration after anti-CTLA4 and anti-PD-L1 treatment in the 4T1 mammary tumor model. On the y-axis is a list of cohorts that match the query and follows a similar naming convention of tumor model, study ID, and treatment condition, followed by the number of samples in the cohort. The x-axis displays the selected immune cell infiltration level inferred by the selected algorithm, noted within the parentheses. Baseline control samples are colored in gray, non-responders are in orange, and responders are in green. We see that only the young mice responded to ICB treatment in this 4T1 model, and there was only a significant increase in CD8 positive T cells after anti-CTLA4 treatment in the young mice, but not with anti-PD-L1 treatment. You can compare the inferred infiltration level from other algorithms by selecting from the drop-down and resubmitting the query. Compared to TIMR, the EPIC algorithm does not estimate a significant difference in CD8 positive T cells after anti-CTLA4 treatment in the young mice of this cohort. From the data download module, you are able to download all our annotated sample metadata and matrices of gene expression and inferred immune cell infiltration. The documentation module provides an overview of how we collected and processed sample data in TISMO. Thank you for watching.